Second component of bacterial cell which is found external to its cell wall is fimbriae and pilus. There is a difference between fimbriae and pili. Both are found attached external to the bacterial cell wall. Their length is comparatively very less as compared to the length of bacterial flagellum. So pili, singular, it is called as pilus. These are the filamentous structures. These are hollow like flagella, and they are nothing to do with motility, but it is in general. This statement is written in general, but in few cases, in case of twitching type of motility, these pili or fimbriae are also involved on the solid surfaces. It is called as gliding type of motility. So gliding type of motility is not because of flagella. It is because of these appendages like fimbriae and pili. These appendages are thin, short and straight, and the number is more than flagella. These are usually called as fimbriae, singular fimbria, but there are differences between fimbria and pilus. These differences are on the basis of structure and function. A cell may be covered with up to 1000 fimbriae. Fimbriae are slender tubes composed of helically arranged protein subunits, and these are about 3 to 10 nanometer in diameter and up to few micrometers long. But fimbriae are shorter than pili. Pili are longer. There are few bacteria which contain these pili. They are called as sex pili, and usually their number is less as compared to the number of fimbriae. Number of pili is less. So these pili are involved in sexual reproduction, and therefore they are called as sex pili. These are also the hair-like appendages like fimbriae, but pili are often larger than fimbriae. Structurally, they are larger, usually 9 to 10 nanometers in diameter. Fimbriae are of 3 to 10 nanometers in diameter, and these pili are generally involved in the transfer of plasmids from one cell to another cell. The process is called as conjugation. The protein that makes these pili are called as pillin proteins. So this is how we can compare the structure of pili and fimbriae. So fimbriae, you can see these are the shorter appendages and their number is more as compared to these longer appendages which are called as pili. So this diagram shows clearly the difference between these appendages like pili are longer as compared to fimbriae, and this is the flagellum. Now the functions of these structural appendages. Let us see the function of pili. It is a type of sex pilus. It is also called as F pilus. F stands for fertility. So these pili are involved in sexual reproduction of bacteria called as conjugation process. So conjugation process is the sexual reproduction in bacteria in which the genetic material is transferred from one bacterial cell to another bacterial cell, and the genetic material which is transferred, it is called as F plasmid or F factor, fertility factor. This F plasmid or genetic material is present only in donor bacterial cell, which is then transferred into other bacterial cell, which is called as recipient cell. The process is called as conjugation. During this conjugation, first the donor bacterial cell, which contains this F plasmid, possess this pilus. With the help of this pilus, this bacterial cell make contact with recipient cell. So initial contact between tip of pilus and the recipient cell takes place. Then this pilus it contracts and brings the two cells closer. When two cells come closer, the cell wall of these two bacteria, that is bacterium, donor cell and recipient cell, the wall to wall contact is formed, then the cell wall is dissolved and there is formation of a channel or a bridge called as conjugation bridge. This conjugation bridge involving the fusion of cell envelopes. Through this conjugation bridge or through this channel, the DNA, that is F plasmid, present in donor bacterial cell is transferred to recipient cell, while the one strand of this F plasmid is transferred to recipient cell through this conjugation bridge. But it is not through the pilus itself. So the function of pilus is just it brings the donor cell and recipient cell closer for conjugation process. This is the electron micrograph. Here we can see this is the donor cell which contains these small appendages. You can see very small appendages. These are fimbriae, and you can see this long one, this long one, this is called as pilus. So this is the pilus which binds to this recipient cell. Then this pilus it contracts and this recipient cell and donor cell they come closer. This is how the process of conjugation occurs. You can see this is the donor cell. This is chromosomal DNA of donor cell and extra chromosomal DNA which is called as plasmid. In this case it is fertility plasmid. It is a double stranded DNA again which possess this bacterial cell possess this sex pilus, and this is recipient cell having its own chromosomal DNA. So this pilus it binds to this recipient bacterial cell like this, and then this pilus it gets contracted. As a result of contraction, this bacterial cell and this bacterial cell they come closer like this. After coming closer, at a particular point the cell wall envelope dissolves and it leads to formation of a conjugation bridge. Now one strand of this double stranded DNA it enters into through this conjugation bridge it enters into the cytoplasm of the recipient cell. While it is entering, one copy of this single strand is prepared or it is manufactured by using DNA polymerase. So DNA polymerase it synthesizes a new strand, and then with the help of replication machinery and transport proteins like relaxosome and transferasome. These are the proteins which are involved in the transfer. So DNA polymerase, using DNA polymerase, new strands are synthesized, and these strands are then transported to the recipient cell's cytoplasm. And the final result is like this. This donor bacterial cell, due to the replication, due to the synthesis of DNA through DNA polymerase, double-stranded DNA structure is retained and one copy is transferred to this recipient cell. Now this recipient cell has F plasmid. So in this case, this F negative cell, this was F negative, now this is F positive. Now it will start producing this pilus, and this is ready to transfer its genetic material to the next bacterial cell. This is how there is generation of new donor cell. This is the process of conjugation in which these pili are responsible for binding to recipient cell and contraction, so that two cells come together for the transfer of plasmid. So this is how this F pilus, it helps in the process of conjugation. The other functions of pili are adhesion to the surfaces. For example, certain bacteria which are responsible for infection in humans, with the help of these pili, these pathogenic bacteria attach to our body cell linings, like in respiratory tract, intestinal tract, and genitourinary tract, as well as it helps for the binding to other host cells. So this adhesion prevents these bacterial cells from being washed away by the flow of our mucus and the movement of saliva and other body fluids, and helps the bacterium to remain attached to the body cells, and that helps to colonize the mucous membranes. So they remain attached and they can multiply there, and this is how it is responsible for the progress of infection, and that permits the start of infection. So these acts as pathogenicity factors. So it helps in the start of pathogenesis, start of infection in case of pathogenic bacteria. There are some bacterial viruses, that is bacteriophages. So these bacteriophages attach specifically to the receptors on sex pillars. So these sex pili, they act as a site for the binding of viruses, and that is also the beginning of the reproductive cycle of these viruses. So viruses use these pili as a site for binding or receptor. Then fimbriae are responsible for binding, and not only binding, these fimbriae are also involved in twitching type of motility. Twitching type of motility that occurs on solid surfaces, which is called as gliding motility, and this is seen in case of bacteria like Pseudomonas aeruginosa, Neisseria gonorrhoeae, and some strains of E. coli. So this movement is with the help of these short appendages. These are intermittent jerky motions, and these movements are also seen in case of myxobacteria, where they are responsible for movement which is called as gliding motility. So this diagram we have already learned. So with the help of these fimbriae, these bacterial cells can move. You can see these are fimbriae, and these fimbriae, they bind to the solid support, and these fimbriae, they contract and retract, and as a result of that contraction and retraction, these bacteria are able So this contraction and retraction, these bacteria are able to move ahead. So there is contraction and retraction. So this is how fimbriae helps in this twitching type of motility, that is gliding motility on the solid surfaces.